Alright guys, welcome to the Exam 1 review of Anatomy and Physiology 1. I have pre-recorded this just in case there are any issues, so you guys can have it. Alright, let's get started. So what is the anatomical position? So this lady right here is in the anatomical position. She's standing straight, hands to the side, palms out, facing towards you, the screen, and then the thumbs are facing away from her body. That is known as the anatomical position. And then the body planes, you have three. You have the sagittal plane, which splits you into left and right. Sagittal, left and right. Then you have the frontal or coronal, which splits you front and back. And then you have the transverse cross section, which splits you up and down. Okay. Now to the side here, we have these abdominal pelvic regions. Okay, they're split into, there are 9 different regions. Okay, you have the hypochondriac on either side, so the right hypochondriac, left hypochondriac, and then you have the epigastric region up top. Then in the middle row, you have the right lumbar, the left lumbar, and then the umbilical. And then on the bottom, you have the right iliac and the left iliac with the hypogastric region on the bottom. So it's going to be important to know these terms, what anatomy and physiology are. Okay, anatomy is the study of internal and external structures of the body. And physiology is the study of how living organisms perform their vital functions. So make sure you guys go over that. Okay. So anatomy and physiology are closely related. Now, you guys are going to need to know the different types of anatomies. There's the systemic anatomy, which is the study of the general form and the superficial markings. You have regional anatomy. Okay, it focuses on anatomical organizations. Then we have systemic anatomy, which studies the systems, the cardiovascular, respiratory. Then we have developmental anatomy, which describes the form that occur between conception and physical maturity. And then you have clinical anatomy, so medical, radiographic, etc. All of these are grouped together as macroscopic anatomy, things we can see with the eyes. Next, we have microscopic anatomy, which has to do with the cells. We have cytology and histology for that. So what is homeostasis? So homeostasis is the ability of the body to maintain stable internal conditions. So that has to do with temperature, fluid balance, pH levels. And the interesting thing about this is that these remain constant despite the changing environment that we deal with. So when it gets cold, our body works to maintain that temperature of 98.6 degrees Fahrenheit. So how does the body control homeostasis? We're going to take a look into that in the next slide. Okay, this is how it is controlled. We have the receptor, okay, that detects the change. Then we have the afferent pathway, so that information is taken to the brain. The brain decides what to do, Then it sends it off, that signal, that information, down the efferent pathway, which goes to the effector. And the effector is basically a muscle, a gland, whatever it is that changes in response to the variable or the receptor over here. Okay, so we're gonna go back here to answer the question of what is a stimulus versus effector. So we know that the body controls homeostasis through negative feedback mechanisms and positive feedback mechanisms. Okay, and the stimulus or the receptor here detects the change and the effector makes the change if necessary. Okay, so that's the muscle, that's the gland. So let's say you're hot, your body senses it, it goes to your brain. Your brain is like, all right, we need to sweat a little bit to maintain that body temperature. So the brain sends the signal to your sweat glands, you start to sweat, body temperature is maintained. Okay, we have negative feedback mechanisms that help control homeostasis, and we also have positive feedback mechanisms that help control homeostasis. So negative feedback causes the variable to change in the opposite direction. So if your body temperature is going down, the negative feedback will cause it to raise, to go back up. If your body temperature is going up, the negative feedback mechanism will cause it to go back down. Now for the positive feedback mechanism, the variable accelerates the change in the same direction. So for this, an example is clotting, or yes, exactly, blood clotting. Once your body detects there's a bleed, it sends the platelets there. So that platelet response sends even more platelets to the site. Even more platelets attract even more platelets. So that continues the mechanism to clot the blood. Okay, now, what are the levels of organization? We're going to take a look at that into the next slide. And then we're also going to identify the organs located in the cranial, thoracic, abdominal, and pelvic. So we have the cranial cavity up top here. We have the thoracic cavity here that houses the lungs, the heart, the esophagus. Then we have the abdominal pelvic cavity, which is this big thing right here. And this houses the rest of our organs. So stomach, intestines, liver, kidneys, our pubic area as well. So gonads, testes, ovaries, all those sex organs as well are inside the abdominal pelvic cavity. So here we have the levels of organization. We start with an atom that becomes a molecule. This molecule either becomes a macromolecule, so something like a protein, a carb, or a lipid. Then we go to the cellular level, where those become cells, and then those cells become tissues. A group of tissues together become organs, and then the organs become these different organ systems here, which we will go through one by one, because he wants you to learn these different systems. We're just going to go through their basic information, nothing too detailed yet. So first, we're going to start with the integumentary system, our skin. So this is the major organs are the skin, hair, sweat glands, and nails, and its function is to protect us from the outside world. Next, we have the skeletal system. The major organs there are the bones, cartilage, bone marrow that produce our blood cells, and in its function is to provide support and protection. Stores calcium and other minerals, and it forms blood cells like mentioned. So the bone marrow does this. Oops. Next, we have the muscular system. So that has to do with skeletal muscles, associated tendons. This to provide movement, also protection. A lot of these things are there to provide protection for our internal organs. Next, we have the nervous system, the main control system of our body. The organs there are the brain, spinal cord, peripheral nerves, and the sense organs. Its function is to direct an immediate response to stimulator, and it sends that out to the effector to make the necessary changes. Next, we have the endocrine system. The major organs there are the pituitary gland located in the brain, the thyroid gland, pancreas. Okay, and this is just basically deals with hormones and communication with the body, and this is also how metabolism is used through the endocrine system. Okay, the endocrine system basically controls your metabolism. Okay, then we have the cardiovascular system, which is pretty straightforward. Heart, blood vessels. What does it do? Just pumps blood and makes sure we stay alive. Then we have the lymphatic system. The major organs there are the spleen, the thymus, the lymphatic vessels, tonsils, and this is basically our defense system. Okay? Our immune system works with the lymphatic system. Next, we have the respiratory system. The major organs there, again, this is pretty straightforward. We have the lungs. Inside the lungs, we have the bronchi, then the alveoli. Our sinuses are part of it, the larynx, our trachea, the voice box. And again, this is just to provide oxygen to the body. We breathe in oxygen, we breathe out carbon dioxide. Next is the digestive system. All of these organs are teeth, tongue, our pharynx, which is the throat, esophagus, stomach, small intestine, large intestine. And this is just to process and digest food, absorb water, absorb any other nutrients that our body may need for later use to function. Then we have the urinary system. This gets rid of waste from the blood. So the organs involved are the kidneys, the ureters, which is the tube that leads to the bladder, the urinary bladder, and then from the bladder we have the urethra, which is the tube that the urine exits out of. Next, we have the reproductive system of the male. The organs there are the testes, and the function is to produce male sex cells. Okay, all for these all levels of organization, don't worry about them too much, okay? Just know the basic idea of them, their function. You don't even have to know all of the major organs, just a couple. All right, next we're getting into the chemical level of organization. So what is a compound? It is a substance formed from two or more elements chemically linked. What is a molecule? It is a group of atoms bonded together by chemical bonds. So the difference between them is this. The molecule is usually described as any atom that's connected by chemical bonds. So it can be two oxygen atoms that make an oxygen molecule. Okay. Then we have a compound is a molecule made up of atoms from different elements. So that's the difference between the molecule and a compound. Molecule can be an atom from the same element or different elements, but a compound is atoms from different elements. Then we're going to get into mass number. This is just the number of protons and neutrons in the element. Okay? And the way you calculate mass number is to just go on the periodic table, find the number right here, and this is your mass number. That's basically it. Now the subatomic particles are pretty straightforward. They are electrons, protons, and neutrons. Electrons are negatively charged. Protons are positively charged. Neutrons have no charge. Okay? Now the rules for the electron shell are this. Every single element has a certain number of electrons. Okay? Now the last shell likes to be filled with 8 electrons. So if we have an element that has all of its electrons filled in the outer shell, it's stable. If an element does not have all 8 electrons in the outer shell, it is considered unstable and will most likely get into a chemical reaction with another atom or another element to fill those orbitals or that last shell with electron to make it more stable. Okay? So again, what are the rules for the electron shell? I've kind of said it already. It is the atoms with the outermost electron shell filled are stable and unreactive. So these are the noble gases. So helium, neon, all of those, they're all very stable and they do not like to react because they're happy. They have all 8 electrons filled in the outer shell. It's the ones that do not have all of them filled that are unstable and reactive. Now, it doesn't mean unsafe. It just means that they want to have their shell to be filled with 8 electrons. Now, we talked about mass number, that we said that it's the number of protons and neutrons in the atom. So an isotope is basically another form of this element, except it has a different number of neutrons. So let's say we have carbon 14, right? It has 14 protons, 14 neutrons. Now let's say carbon 15, for example, still has 14 protons, but now it has 15 neutrons. So it has an extra subatomic particle in there, making it heavier than the regular element, thus making it more reactive. Okay? So an isotope of the same element has the same number of protons, but a different number of neutrons. So how do we stabilize the chemical behavior of an atom? Pretty simple, just have it react with another element so its electron orbital can be filled with 8 electrons. So a polar covalent bond is just a covalent bond that has an unequal sharing of electrons, which is shown right here. Okay, this element here is attracting more electrons to its side than this element here. Okay, a covalent nonpolar bond just means that there's equal sharing of electrons between the two elements. And that's the difference. So the polar covalent, they have unequal sharing of electrons, and the nonpolar covalent, they have equal sharing of electrons. So sodium bicarbonate, let's talk about that a little bit. So it is NaHCO3. Okay, we talked about how our body maintained pH levels through homeostasis, and this is one of the methods that is used. Well, our body doesn't really use sodium bicarbonate, but it uses its anion, HCO3 negative. So this binds with hydrogen ions, which makes something more acidic. Okay. H plus HCO3 bind together to make H2CO3, which brings down the pH or the acidity of the blood. So our body uses this right here to maintain normal pH levels. So we're going to talk about water now. We have the properties of water. We have solubility. It's very reactive. It has a high heat capacity, meaning it's able to hold a lot of heat. And it's used as lubrication and cushioning in the body. Okay, water has amazing properties that make it a very strong solvent, make it a good medium for reactions to happen, and it holds a lot of heat. So catabolic reaction is something that makes a larger molecule into a smaller molecule. Okay, or otherwise it's just breaking something down. Anabolic reactions build up compounds. So you start off with a smaller molecule, there's energy, and then you end up with a larger molecule. So this is a little bit about the HCO3 I was talking about. This is how our body maintains its level pH through HCO3. So this hydrogen proton is what causes the acidity. The more of the hydrogen proton we have in the solution, the more acidic it is. So HCO3 combines with it to make carbonic acid to reduce the acidity in the blood. So we're going to talk about the types of chemical bonds right now. We have ionic bonds. By the element is either losing or gaining one electron, like literally it loses it or it gains it. So one of them is positive, and a positive, and then the other is negative. Now, covalent bonds, the electrons are not lost or gained, they're just shared. Like we discussed, the polar covalent bond has a unequal sharing of electrons, and the nonpolar has a equal sharing of electrons. And then with covalent bonds, we also have single bonds, we have double bonds, and we have triple bonds. And also we have polar and nonpolar covalent bonds. Next, we're going to talk a little bit about proteins. So this first exam is just basically a little bit of general information about everything. So you gotta know some chemistry with those bonds. We're talking about proteins now. Before we were talking about levels of organization. So it's just a little bit of everything. The next couple exams, or the next exams moving forward, will just be one topic that you'll focus on. So it won't be all over the place. So what are amino acids? So amino acids are just hydrocarbon chains, meaning they're just chains of hydrogens and carbons, okay, with an amino group, which is this part right here, the NH2, and a carboxylic acid group, which is the COOH. Okay? All amino acids have basically the same general structure. They have the central carbon right here. They have the amino group, which is why it's called an amino acid. It has the carboxylic acid group. The only difference between the amino acids are the side chains right here, the R side chains, which is just an abbreviation. So we have 20 essential amino acids, okay? Amino acids. The only difference between these 20 amino acids is the side chain right here. Besides that, they all have the carbon central atom. They all have the amino group. They all have the carboxylic acid group. The only difference is this side chain right here. Each different amino acid has its own special side chain, consisting of either carbons or hydrogens, sulfur sometimes. It could be carbon, hydrogen, oxygen, just different combinations. So the way amino acids bond to one another is through a peptide bond. What happens is the amino end of one attaches to the carboxylic acid end of the other. Okay? So they just attach side by side like this. So the amino group attaches to the carboxylic group, the amino group attaches to the carboxylic group. And these A's are just amino acids, different amino acids. They're literally attached side by side like this. Okay? Now the structure of the amino group, we just talked about it. They have actually, no, we didn't talk about it. We talked about the structure of the amino acid. Now the structure of the amino group is just this. NH2. Okay? One nitrogen atom and one and two hydrogen atoms. So proteins are basically chains of amino acids. If the amino acid chain is long enough, then it is enough to be a protein, or what's otherwise known as a polypeptide. Okay, now we're going to talk about how our body uses proteins. So we use proteins for a wide range of things. We use it for support, for movement, okay? Our muscles use proteins to contract and relax, transporting stuff. So it could be liquids, it could be molecules that the cell needs, and the protein functions to transport it from one side of the cell to the other, or maybe it transports it outside of the cell. It's used as a buffer to maintain physiological pH. It has metabolic regulation functions, okay, and it also is used for defense. Keratin on our nails, it's a tough protein that's there for protection. Now we're going to get into the structures of proteins, okay, which is going to be the next couple of slides. So there's a primary structure, secondary, tertiary, and quaternary. So let's get started. So the primary structure is simple. It's just a sequence of amino acids. That's it. So if you have just a chain of amino acids, then that's the primary structure. And this is just connected to just simple peptide bonds. Remember we talked about how the amino group connects to the carboxylic acid of the other, and they're just attached side by side? Well, this is the primary structure. Next, once this chain gets long enough, we have the secondary structure, where the hydrogen bonds react with the hydrogens on another amino acid. So this A2 right here is reacting with this A6 right here. This fourth amino acid is bonding with this 8th amino acid right here. So because of this chemical attraction, this creates two structures, either an alpha helix, which is shown right here, okay, just how a regular our DNA looks, how we're used to seeing it. And then we have on the next slide something called a beta pleated sheet. So there are two secondary structures, alpha helix and beta pleated sheet. Okay, don't forget the beta pleated part. Now this structure arises just from the reactions of the amino acids on the same chain. That's it. Next, we get into the tertiary structure. So all of these different structures of the secondary, we have the beta pleated, the alpha helix here, beta pleated sheets right there. All of that become the tertiary structure. It is just a complex coiling and folding that gives the protein its final tridimensional shape. Most proteins are in this tertiary structure right here. Okay, so this is an active functioning protein now. And lastly, we have the quaternary structure, which is just different tertiary structures combined together to form this mega protein structure complex here. Usually these are antibodies, they can be enzymes, and etc. Okay, this is just a summary overview of the primary, secondary, tertiary, and quaternary structures. So the primary structure is the sequence of amino acids along the length of a single chain. The secondary is the arrangements of the amino acids due to the hydrogen bonds between different parts of the chain, which makes the alpha helix or the beta pleated sheet. Now, eventually, these chains become long enough where the alpha helix and the beta pleated sheet start convoluting and twisting and coiling into this final tertiary structure that is a functioning protein. Now, if needed, the protein can get into this quaternary structure, which is just a bunch of different tertiary structures combining to form one big complex. So what is a glycoprotein? A glycoprotein is an oligosaccharide chain covalently attached to protein. An oligosaccharide chain is just a carbohydrate chain. And a proteoglycan is a core protein covalently attached to another carbohydrate chain. Okay? Now their locations are different. Proteoglycans are on connective tissues for support, and glycoproteins are used at the cell surface for signaling and recognition. Glycoproteins are not that much carbohydrate content. They don't have that much carbohydrates in them, as opposed to proteoglycans, which the majority of the structure is a carbohydrate. So glycoproteins are used for cell signaling and recognition, and proteoglycans are used in collagen to form cartilage. So the function of an enzyme can be your protein, is to speed up the reaction, okay? An enzyme has an active site where the substrate or the molecule that is undergoing the reaction attaches so the enzyme can fold and create the necessary product that is needed. So how does an enzyme interact with a substrate? It does this through the enzyme substrate complex. The enzyme comes along, it the substrate is next to it. The substrate, if it attaches, if it fits, I mean, sorry, it's kind of like a lock and key. If it fits, it attaches to the enzyme. The enzyme, boom, right here, it attaches. The enzyme changes its shape. Okay, and then once it changes its shape, it combines these two together, it releases it, and the enzyme goes back to its regular shape. So this is an anabolic reaction because it's building something up. This is just some more information about an enzyme. Sometimes they have cofactors. So they need these ions right here before it's able to bind to the substrate. And sometimes they have coenzymes, which is something that is not a protein, but it needs to bind to the enzyme before the substrate can bind to it. So we're going to talk a little bit about carbohydrates right now. So these are the different types of sugars. We have monosaccharides, disaccharides, polysaccharides. Monosaccharides are only 3 to 7 atoms of carbon. Disaccharides have more than that. Disaccharides are just basically two monosaccharides bonded together, and that just makes a disaccharide. That's it. Usually it's through a dehydration synthesis reaction. And that's how you construct disaccharides. You take two monosaccharides, it's usually a dehydration synthesis reaction, and that creates the disaccharide. So some carbohydrates, we have glucose, fructose, we can have starch, chitin. All of these are carbohydrates. Some are simple, like these are simple carbs. And then we have complex carbs like starch found in potatoes. So the cells that store glucose or fat are called adipose cells. So starch is an example of a carbohydrate source. They're found in plant. Glycogen is the storage of glucose. In the previous slide, the storage of fat was for adipose. The storage of glucose is glycogen. Our muscles have a lot of this glycogen, the liver as well. So that fight or flight reaction that we were talking about, or that the professor was talking to you about, has to do with this glycogen right here. When we're faced with danger, we don't have time to eat. So our body usually uses this glycogen reserves right here to release the glucose so our body can use the energy. Chitin is also another complex carb, and these are found in the external skeletons of arthropods. Now we're going to get into lipids. Okay, these are the different types of lipids right here. Fatty acids, eicosanoids, glycerides, steroids, and glycolipids. An example of a glycolipid, we have phospholipid. An example of glycerides is the triglycerides, which is what our body uses for energy. This is the fat in our body for energy. We're going to go into the function of each and where they're found. So for fatty acids, we're going to first talk about the function of it, and then we're going to get into these questions up top right here. So we either have saturated fatty acid or an unsaturated fatty acid. The saturated have no double bonds in them. They're just a group of carbons and hydrogens. Unsaturated fatty acids, they do have a double bond, and these tend to be more unstable in the body. So the difference between a saturated fatty acid and unsaturated fatty acid is the double bond, okay? Which makes it more unstable and basically unhealthy in the body. So these are located in the food, and fatty acids are used for structure as well in the body. So we have eicosanoids, which is a group. Prostaglandins and leukotrienes are eicosanoids, okay? And these are just basically lipid compounds that are released by cells to control cellular activity, okay? So this is just basically for cell signaling and communication. And same thing with the leukotrienes. They're also involved in regulating the immune system. So again, just basically communicating and defense. These are what the eicosanoids do. Next, we have the glycerides. They're the most common type of lipids. These are also found in structures of the cell, and their function is just to provide protection. And the triglycerides themselves are used as energy. So not protection, but energy. So we have different types of glycerides. We have a monoglyceride, a diglyceride, or a triglyceride. And that just has to do with the number of chains sticking out of the body. The monoglyceride has one chain, diglyceride has two, triglyceride has three chains. So what is needed to make up a triglyceride molecule? Well, you need the glycerol backbone. Sorry, that's the glycerol backbone and the fatty acid chain. So this one has three fatty acid chains, which makes it a triglyceride. So if it had one fatty acid chain, it would be a monoglyceride. If it had two fatty acid chains, a diglyceride. So energy, insulation, protection. Next, another common type of lipid is we have steroids. These are basically for structure. They provide communication with the rest of the body as well. These are found on cells as well. Okay, these are the principal steroids. Cholesterol, very important for stability and structure. It maintains our phospholipid bilayer or the cell membrane. Steroids can become sex hormones or they can become corticosteroids, which cortisol is involved in fight or flight. So the function of steroids, I said that the cholesterol is to maintain the integrity of the plasma membrane. Again, creates sex hormones for regulation of sexual functions, regulates metabolism and neural balance, and they are required for digestion and absorption of fats. And then our last bit of lipids, we have phospholipids. These are found in the cell membrane. Okay, we have one head right here with two tails. This is a phospholipid. Head with two tails. So our cell membrane is two layers of this. So this is our cell membrane. Just a bunch of these phospholipids. So this right here is just a summary of the lipid types and their primary function. So fatty acids, primary function is energy source. Eicosanoids, we said that it was just for messages and to regulate local cell activities. Glycerides, again, are for energy sources, for physical protection. Steroids, they provide the structure of the cell. The phospholipids, they also provide the structure of the plasma membrane. So why are omega-3 fatty acids good in the human diet? Well, because they reduce the risk of heart disease, rheumatoid arthritis, and other inflammatory diseases. So it reduces inflammation in the body. Last but not least, we're going to talk about DNA and RNA and what the differences between them are. So this says a components of DNA slash of DNA. We're going to put slash RNA. Because really they're practically the same thing, but we're going to get into the differences right now. So they both have a sugar right here. They both have a nitrogenous base, N base, and they both have a phosphate group. Now the difference lies with the sugar and the base. DNA sugar is a deoxyribose, while RNA is just a simple ribose. It looks something like this right here. This is the sugar. Okay? The base is also different. Now when we talk about bases, we're talking about the purines and pyrimidines. The thymine, adenine, guanine, and then cytosine. These are the bases found in DNA. The only difference between DNA's base and RNA's base is that RNA does not have thymine. Okay, it just has uracil. So for RNA, instead of the thymine, it's canceled out and it has uracil. But the rest of these bases are the same. Another difference is that DNA is double-stranded while RNA is single-stranded. So it means that DNA forms a double helix and RNA does not form a double helix. Okay? So let's review these questions once again. What molecule makes up DNA? What molecules make up RNA? We said the nitrogenous bases, the pentyl sugar, and the phosphate group, which is right here. So the molecules that DNA and RNA have in common are the phosphate group, honestly, because the pentyl sugar is different. We said deoxyribose for DNA, oxyribose for RNA. But there are some bases that are similar, right? We said the only difference between DNA and RNA is the thymine and uracil. Well, they both still have guanine. They both still have adenine. And then they both still have cytosine. Those are the molecules in common. And then what molecules do they differ in? We just said that is thymine and uracil. And of course, the pentyl sugar base. because DNA has deoxyribose, and then RNA has the regular sugar ribose. Oh, that's it, guys. I hope you guys were able to watch this. If you guys have any questions, please feel free to reach out to me. I apologize that we're not able to go live this time. Something came up and I had to pre-record it. But next time we will be live. But please feel free to reach out to me. I'll make sure to check my email over the weekend to answer any last minute questions. All right, good luck studying.